Hello everyone, this is your instructor Kwame Shen. This is the last part of our lecture on eukaryotic classification, and for this lecture we will look at insects as a continuation of the phylum Arthropoda, and then also look at the kingdom Fungi. So insects, unlike arachnids, have 6 legs. So they have, in terms of the tagma, they have 3 body divisions: the head, the thorax, and the abdomen. There is a large variation amongst insects. They are one of the most diverse groups of organisms on Earth, and so there's variation in terms of the type of wings, the number of wings, the sizes of their legs, from the front legs to the hind legs, the type of mouth parts that they have, which are adapted to their feeding habits. So examples will be the fleas, the mosquitoes, kissing bugs. So if we look at the fleas, for instance, because of their medical importance, fleas are small, they are flat. We say they are flattened on the side and not dorsoventrally. So this is what I mean. You could have flatness in this direction of an organism in respect to the earth, and then you could have one that is flattened in such a direction with respect to the earth. Fleas are flattened in this manner. They are wingless. Most of them would be parasites of rodents and birds, where they may feed on their blood every now and then, or they may have to feed on their blood continuously in the context of body larvae. And then they may carry some pathogenic organisms, and that's why they are important to us, like tapeworm, Yersinia pestis, which is the causative agent for plague, and some other bacterial, protozoans, and helminthic diseases. Fleas, in terms of their classification, is not really fixed. Generally, when it comes to classification of organisms, especially when it comes to the arthropods, there are a lot of changes that are always taking place. But representatives of the flea would be Pubescens irritans. This is the picture of Pubescens irritans. But you can see how the morphology of the legs are, that the hind legs are longer and more powerful than the forelegs, and it helps it in springing. So it depends on the organism's lifestyle. Then you have the body larvae, which causes pediculosis. Pediculosis is an infection of body larvae. They lay their eggs on or near the seams of the clothing. So typically, if you want to check for them, you want to turn the clothing inside out and look at the seams to see whether you would actually see any indication of a contamination with body lice. They feed on blood, and they have to feed on blood continuously. They're endemic worldwide, and you typically will find them in settings where there is crowding. So you're looking at homeless settings, refugee settings, and stuff like that, where there isn't much hygiene and there isn't much laundering or regular baths taking place among the individuals. And so the scientific name is Pedunculus humanus corporus. The corporus refers to the body. Humanus, it's a human body louse. And you can see one here that's engorged with significant amount of blood. In terms of the life cycle, there is the nit, which is the egg. The eggs of lice are called nits, N-I-T. There's a nit, there's a nymph, and there is the adult. This is an example of incomplete metamorphosis in terms of the changes that takes place in the life cycle of the organism. When we looked at the arachnids, you see that they have the eggs, they have the larva, they have the nymph, then they have the adult. Anyway, so these have three body forms. So the nits are lice eggs. The nymphs will mature into adults 9 to 12 days after hatching, and they have to feed on blood. The adults also have to feed on blood. And so the moment they fall off from their hosts, they literally die. Then we have the crabs, Therus pubis, the pubic crabs, the pubic lice, and they cause the disease or the infection theriasis. Theriasis example is what you have in the picture, where you can see that the pubic area is infected with crab. It's transmitted mainly through sexual contact. However, you may find crab larvae in the eyelashes, or in the eyebrow, the armpits, where you have hair follicles that have very thick bases. It can be treated with 1% permethrin lotion, or mouths containing permethrin, or peperonyl butoxide. So there are treatments that can be administered for this. Then for fungi belong to the eukaryotic groups. They are non-motile. They have a polysaccharide wall. And in terms of how they obtain nutrients, they will secrete enzymes externally, digest dead organic matter, and then absorb the nutrient into their body. Generally, they are parasitic forms, however. So we call them saprophytes. So they are saprophytic in their feeding habits, because they feed on dead organic matter. There are two types. You have the unicellular forms, the yeast, and the filamentous multicellular forms, the molds. In terms of their life cycle, you may have some that will show both the yeast and the mold life cycle. Those are known as dimorphic. So those are dimorphics. And then you have the macrofungi. The macrofungi would be the mushrooms, the puffballs, and the shelf fungi. So all the three that I mentioned have been represented here. So these are the yeast, these are the molds, and then these are the macrofungi. Fungi would have cell structures that are called hyphae. So they form these filamentous cell structures called hyphae. A collection of the hyphae is known as mycelium. Now, based on how the mycelium appears, the mycelium may be lightly pigmented, or typically what we describe as unpigmented. So there are no pigments in the hyphae, and those are known as monolaceous. And then you have the demaciaceous. Those are the ones that have pigments in them. The hyphae could also have cell walls. So there are some hyphae that have septum. There is divisions in the walls of the cells, and those are known as septate hyphae. And there are others that are just continuous. There are no divisions. Those are known as non-septate or coenocytic. So they are continuous. They have a very complex life cycle. They may have sexual and asexual forms. They may form sporangia, gametangia, or zygotes, depending on the type. They are categorized based on, fungi are categorized based on the type of spore that they form. So they may form spores at the end of their hyphae, and these spores may not be in any capsule. They're not covered. They are naked spores, and those will be what we describe as Cornelia. They may also form spores by budding, and typically that's what you see in the yeast. So you have a parent cell, and then there is a budding of the parent cell, and those are known as blastospores. You may have those that will form the spores from the hyphae that become fragmented, and those are known as arthrospores. And then you have the chlamydospores, which are basically spores that have a very thick covering around them. They may be formed at the edges or the tips or somewhere in the middle as well, but they are known as the resting stage. They are characterized by thick walls. If you can classify fungi based on the type of hyphae that they have and the spores that they form. So we have the division Glomerulomycota, Ascomycota, and Basidiomycota. And so if you look at the spore formation and the septation, whether it has a cell wall or not, Glomerulomycota would have members of Zygomycota, and they are mainly terrestrial. They don't have a septate. So these groups do not have a septate. And they are non-motile. They can form asexual spores, known as sporangiospores, or they can form sexual spores, known as zygospores. And then you have the division Ascomycota with members Ascomycetes. They will produce a sac-like structure in which you will find their spores. The structure is known as ascus, and they produce spores sexually, so meiotically. An example would be yeast. Then you have Basidiomycota. They also have septates, so they have septates. They'll produce a structure known as basidium, on which the spores are contained or carried, and they will also produce spores sexually by meiosis. We'll look at then some medically important fungi. So Candida albicans is a very important fungi that has important medical implications. It can cause oral thrush, vaginitis, or candidiasis in the skin. It's part of the normal flora, so you find Candida as part of the normal microbiota of the respiratory, gastrointestinal, or the urogenital parts of the body. They produce what we call a pseudo hyphae. A pseudo hyphae is formed when there is budding, but then the cells, the chains that are formed fail to break off. So look at this picture here. You see budding here, you see budding here. But for either case, you see that they are still attached to the parent cell, so there is no breaking off of it. So it forms a false hyphae. So there are chains of budding yeast, and they will contain chlamydospores. Then you have Sarcomeres Cerevisiae, which is the baker's yeast. Sarcomeres Cerevisiae is an ascomycote. It has vegetative state. The vegetative state is the state that it's used to absorb the nutrient, and you see all the growth taking place. If you see a fungi on a plate, you're looking at mostly the vegetative stage at that point. You may see some spores, but those will be aerial spores, filaments that will point upwards, and then you have the spores on top of it. If you culture them, they form colonies just like bacteria, and as you can see in the plate there, And these have been cultured and incubated, and the colonies that they form are just like bacteria. They appear larger though. Critically, they are used in the wine and the bread and the beer industry. Then you have the bread mold, the rhizopus species. They are fast growing. They have the color of the salt and pepper. They will form hyphae. The hyaline or the hyphae walls are aseptic, and they will contain rhizoids and sporangiospores. Rhizoids are formed as part of the hyphae that mostly is connected to the substrate. And then you would have these projections, these structures, the sporangiospores, that will have the sporangiospores, the spores which will eventually lead to more cells of the fungi. Examples in terms of representatives, you have the rhizospores, stolonifer, which is a bread mold, the arizos and arizae. So the life cycle of a rhizospores typically would have both asexual and sexual forms. For the sexual reproduction to take place, there are two strains of the rhizospores that have to come together: the donating cell and then the accepting cell. So the donor cell is designated positive, the accepting cell is designated negative. And because you need cells of these two strains to come together, it is not too frequent compared to the asexual stage in terms of the spore production. For asexual spore production, you would have, so these are rhizoids, you have a sporangiospore, and then it will have a sporangium. The sporangium will then have the spores. Okay. Remember, I'm using the words in terms of the suffix for and spore. So the for will be the structure. Sporangiophore will be the structure that will bear the sporangiospore. And so you have several haploid spores that are produced, and then these spores can then generate again into sporangium. However, in the sexual reproductive component of the life cycle, you would have these two strains coming together. Eventually, they will form a zygote, and then the zygote will then also produce a sporangiospore, which will eventually produce a sporangium that will bear sporangiospores. Okay. Another important fungi is Aspergillus. So these are white molds, white ring molds, as you can see in the picture that I have down here in the subset. They produce septates, hyphae, and they are branching in their presentation. So you can see the branches that are taking place. The spores are produced at the ends here, and they are not in any sac or covering. Then you have representatives of them being Aspergillus fumigatus, which is Aspergillus that is implicated in asthma. It's also the cause of aspergillosis. And then you have some that are used in the food industry as well. For penicillin, a very important fungi in terms of the benefits that we get from it, it produces green molds with furrows in them. So you can see the green molds down here, but you can see that there are these lines that runs across it. So these are the furrows. So green molds with furrows in them. They also have they have hyphae. The hyphae have septa or septum, but they are very thin. They also produce branches, just as the Aspergillus, but they have another component known as the metulae. So you see these branches here. They support this other branch that was also seen in Aspergillus, known as the phyalates. Another important examples will be the Penicillin notatum, which is penicillin, the one that is used in the production of penicillin. So I have some supplemental slides here to shed more light on the fungi. They are chemoheterotrophs in terms of how they generate energy using organic sources, and they're then able to produce their own food. So they will secrete digestive enzymes, consume the nutrients from the digestive activities of the enzymes. That is an externally from the cell, and then there are some of them who are parasitic, and those have modified hyphae known as Hostoria. You typically we see a lot more of the historic forms as parasites of plants, and most of them are aerobic. There are few facultative anaerobes. For reproduction, there is asexual reproduction that you see in the yeast that results in pseudo-hyphae, and there are filamentous forms that would also undergo asexual sporulation. They will produce spores such as sporangiospores, chlamydospores, and conidiospores. For the different spores in the filamentous molds, sporangiospores are sac-like, so they will have sporangia, which is a sac that will hold the spores, and they have a stalk known as sporangiospore. So the stalk is sporangiophore, the sac is sporangia, and the spores are sporangiospores. They are found at the ends or the sides of hyphae. So at the ends over here, there's a sporangiospore, there's a sporangium, and the spores are contained there. Then you have the chlamydiospores. The chlamydiospores would have the spores in the very thickened hyphae. So there is a hyphae that is thickened here, and the spores are in here. They may be formed at the ends or in the middle of the hyphae. Then you have the conidiospores, which forms conidia. These are asexual spores that are formed typically at the ends, the tips. They don't have a sac. That's one of the main difference. They are not containing any sacs. So example, when they are formed from the hyphae, they are known as arthroconidia. When they are formed from the parents, there is budding from the parent cell, they are known as blastoconidia. And then when they are chains, example in this case, they are chains that are formed at the ends, then they are known as conidiospores. For the sexual component of their life cycle, there are gametes that have to come together. The donor and the acceptor, positive and negative, they have to come together. So there are three phases of the sexual reproduction, that the production of spores by sexual means. You would have two gametes coming together, the positive and the negative coming together, then they will fuse together. And so this is what we call plasmogamy. So you have the two cells fusing together, and then the two nuclei would then fuse together, and that is karyogamy, to form a 2N. So N and N will come together to form a 2N, and then once the 2N is formed, they will undergo meiosis to form haploid spores again. So that's the life cycle. If we look at that of rhizospores, and in terms of its sexual and sexual, that's the bread mold, an example of that. So you have the sexual life cycle over here on the right. What will happen with the sexual life cycle? You have the two strings coming together, forming N plus N, known as the dikaion. Kaion has to do with the nucleus. So you have two nucleus, the cells are together. The cells are fused, but the nuclei are not fused yet. And then they will then eventually fuse. You have a zygosporangium. A zygosporangium would then produce sporangium. a sporangiospore, where we'll then have a sporangium, and then the spores will be released as well. And once the spores are released, the spores are haploid. When they have the right environment, they will grow and then form mycelium. And the mycelium will also develop sporangiospore and sporangiospores, or the spores will then be produced on the sporangiospore, and then the cycle will continue. So this brings us to the end of our lecture on the classification of eukaryotes.